right, this lecture is about lipids. Just like carbohydrates, lipids have carbon, hydrogen, and oxygen, and sometimes some other elements, but there is less oxygen in lipids than there are in carbohydrates. Lipids include fats, oils, waxes, and steroids, and they tend to be nonpolar. And because we have already learned that water is a polar solvent, they do not dissolve very well in water. The building blocks of fats include glycerol and fatty acids. There is always one glycerol and 3 fatty acids in the kinds of fats we're going to be studying. So we can't really call the building blocks monomers because we do not have a long repeating chain. So it's not really a monomer if you're not building a polymer with them. So we do call them building blocks, but there's only 4 pieces that get put together. So it's not really considered, monomers and polymers. So this is the only section of the biochemistry unit where we really do not have the concept of monomers and polymers, but we do have the concept of building blocks. Now, the first building block we're going to talk about is glycerol. So I'm going to show you the building blocks first, and then I'm going to show you what the whole molecule looks like when it comes together. So the first building block is called a glycerol molecule, and it has 3 carbons in the chain, and it's got 3 hydroxyl groups. Be very careful because students sometimes get glycerol and glycogen mixed up because they both start with the same sound. So unsaturated fats contain fatty acids with double or triple bonds between the carbon atoms. So I will show you an unsaturated fatty acid. Typically, when you look at an unsaturated fatty acid, it's often bent, and you'll notice that there is a double bond between these 2 carbon atoms here. And this is only one type of fatty acid that can be bonded to the glycerol that I showed you on the previous screen. So unsaturated fats tend to be found in plants like olives and avocados and walnuts and almonds. They tend to be liquids at room temperature, so more often they're the oils that you have in glass containers at home. Monounsaturated have 1 double bond in the hydrocarbon chain, and here is our hydrocarbon chain. So would this molecule be a monounsaturated fat, or would it be a polyunsaturated fat? A polyunsaturated fat would have many double or triple bonds within the hydrocarbon chain. The answer to that question is that it is a monounsaturated fat. There's only 1 double bond between carbons. Now, careful, because this group on the end here, this carbon double bonded to an oxygen, bonded to another oxygen and a hydrogen, COOH, this part is called a carboxyl group. And that's in every fatty acid, whether it is saturated or unsaturated. You do not count the double bond between the carbon and the oxygen here when you're counting double bonds to decide whether it is monounsaturated or polyunsaturated. Saturated fats contain fatty acids with all single carbon to carbon bonds. So this is a diagram of a saturated fat, not a saturated fat, a saturated fatty acid. Now be careful because you still have your carboxyl group, the carbon double bonded to an oxygen, but if you notice in the chain, all of the carbons are single bonded. So what do we mean by the term saturated? Saturated usually means full of something. If we were talking about a kitchen sponge, saturated would mean it's full of water. In this case, saturated means full of hydrogens. So if we look back at the previous slide and we look at this double bond, remember that carbon can only make 4 bonds. So 1, 2, 3, 4. If it's making a double bond, it can bond to fewer hydrogens. So if you look at this carbon, it's bonded to mostly to other carbon and only to 1 hydrogen. If you added hydrogen, you could actually break that double bond in the chemistry lab and break the double bond between the carbons and add more hydrogen and fill it. So this one is saturated with hydrogens. You couldn't add any more. There's no double bonds between carbons to break to add more hydrogen. Saturated fatty acids tend to be found in fats found from animals, like the marbled fat in a steak or lard, or actually anything in any dairy product like butter or cream or even whole milk. They tend to be solid at room temperature. And be really careful, again, that double bond does not make this molecule unsaturated. It's part of the carboxyl group. Remember that, again, that carboxyl is that COOH. So here is a picture of a triglyceride. So this is the fat. Those other things that I was showing you, these chains are called fatty acids. The whole molecule is called a fat. Students often get that confused and they'll use the terms incorrectly. So again, these hydrocarbon chains are called fatty acids. The whole molecule, when it's a triglyceride, is called a fat. So its sort of common name would be a fat, and then its scientific name would be a triglyceride. Triglyceride because it's 3 fatty acids and combined with a glycerol. So the tri comes from the fatty acids and the glyceride comes from the glycerol. And we are seeing in this whole diagram an old friend, dehydration synthesis. So we start off with a glycerol and 3 separate fatty acids. We have 3 waters formed, an H and an OH removed, so a hydrogen removed from the carboxyl group, and the hydroxyl, the OH group, from each of these carbons in the glycerol molecule. And we get 3 water molecules because remember, we're attaching 4 molecules together. Therefore, we get 1 fewer water, so we get 3 water. So a triglyceride, also known as a fat, is formed by the dehydration synthesis between glycerol and 3 fatty acids. So remember, we're taking 4 smaller molecules, putting them together to make a larger molecule, hence the term synthesis, and dehydration because water is being removed to be able to get those molecules to come together. Now, of course, when their hydrogen and hydroxide are in the molecules, they're not water yet until they react and are released. So what makes one fat different from another fat is the different combinations of fatty acids. So you always have a glycerol and 3 fatty acids. That doesn't change. But what changes is the length of these hydrocarbon chains. And of course, in order to fit on the screen, these are significantly shorter than real molecules would be. The hydrocarbon chains are typically significantly longer than that, but for the sake of making a diagram that will fit on a screen or in a textbook, they had to shorten them. Sometimes they have double and triple bonds in them, sometimes all the bonds between carbons are single, and there's a very large variety of fatty acids that can be in a fat. So this shows you again the animation dehydration synthesis. And it shows you the water molecules being formed and the new bonds being formed that are, have space now to form because the hydrogen and hydroxyl have been removed. And now we have a triglyceride. This one would be a monounsaturated fat. You see that it has a double bond here. Now, just a little bit of a discussion about which fats are healthy for you to eat and which fats are not. There has been quite a bit of debate back and forth about the health of the different types of fats. But what we seem to have come to agree on now in the scientific community is that it is unhealthy to eat animal fats. Of course, anything in small amounts is not a big deal, but we want to be careful not to eat too much animal fat. And in fact, even the refined oils, like corn oil and vegetable oil, are probably not very good for us. The best sources of fat for our health are typically the monounsaturated fats, but extra virgin olive oil is a healthy source of fat. Avocados and most of the nuts have very, very healthy fats. And of course, the fats found in some fish can be quite healthy. Of course, you have to be careful because these days many fish have heavy metals and pesticides in them. But fish oils are typically your healthy oils. So again, typically we want to be eating plant fats and not really so much the refined plant fats so much as the whole plant foods that have a good source of fat, like avocados. And you know what? You can't really go wrong with guacamole. So this slide, I want to talk about another health issue related to fats. Now, I want to make it clear that we are not going to be asking about trans fats or hydrogenated oils on any of our tests and quizzes. We just thought it was important to add because it's in the news, you hear about it, you read about it, and you should be aware of it when you're shopping and reading labels and purchasing food. So you've heard probably about trans fats before, and you've probably heard they're bad for you. Now, I want to explain what a trans fat is. So remember that when you have your carbon to hydrogen bonds, sometimes the hydrogen will be across from each other. Trans means across, like a transatlantic flight crosses the Atlantic Ocean. Here, the hydrogens are across the molecule from each other. This is the trans configuration. This is called the cis configuration, and the hydrogens on each of these carbons are on the same side, and this tends to make the molecule bend, whereas the trans configuration is straight. Now, trans fats are found in very tiny trace amounts in nature. We actually create trans fats when we take polyunsaturated fats, typically in oil and most often in soybean oil because it's the cheapest. And we take it into the lab and we hydrogenate it, which means we break some double and triple bonds and add hydrogen to it. And for some reason, when you add hydrogens and break triple bonds into double bonds, it tends to add the hydrogens across from each other. And in nature, they're usually on the same side. So it turns out these trans fats are very, very unhealthy for us and lead to terrible health problems and inflammation in our body, and often heart disease at greater rates than even the saturated fats. So the worst kinds of fats are these trans fats that are created in the lab. When we were first making them, we thought they were healthier because we thought oils were healthier than butter, and if we took an oil and made it more solid like a butter, it could be used as a butter substitute and would be healthier. And you're probably familiar with margarine, and margarine is trans fat. Crisco, which you probably have in your cabinet if you're, one of your parents or maybe you is into making pies, because it's very useful for making pie crust. It gives really great consistency. Now, trans fats are used a lot in processed foods, partially because it is cheaper to buy hydrogenated oils and soybean oils and buy Crisco than it is to buy dairy, because remember, with the 90% rule, you have to feed the cows and then milk the cows, and it takes a lot of grazing land to do that. And then also dairy must be refrigerated. Whereas your Crisco, your trans fats, your hydrogenated soybean oils do not need to be refrigerated. So they're very cheap for the large food companies who are making a lot of baked goods. And in addition to being cheaper and not being required for refrigeration, they also create really good texture and what they call in the food industry as mouthfeel. So you do get a really flaky pie crust when you use hydrogenated oils, although you do not usually get quite as good a flavor as butter. So most people who bake pies kind of go 50-50 with Crisco and butter, so that you get the flavor from the butter and the texture from the Crisco. But we really shouldn't be eating trans fats. Now, it's probably not a big deal to have trans fats when you make a pie once a year for Thanksgiving or Christmas, but you should be looking at the labels of the things that you eat every day. Now, food labels can say they're trans fat free if they have less than 1 gram per serving, and guess who gets to decide the serving size? They do. So what you really need to do is look at the ingredient list for the word hydrogenated or partially hydrogenated, and it will then say any kind of oil. So it will say partially hydrogenated soybean oil or hydrogenated soybean oil. And those are the foods that you should avoid. And in fact, in some places, they've actually made it illegal to use trans fats in restaurants. And they often used to use them for frying French fries, and they would use them over and over and over again. And so they have been banned from use in restaurants in certain cities. It's not a federal ban, but it's can be city by city or sometimes state by state. Again, I just want to reiterate, none of the stuff about trans fats or hydrogenated oils will be on the test. I just think it's important when you are shopping to know what to avoid. Now, what I'm about to come back to is another kind of lipid. Now, this is going to be on your tests and quizzes, and in fact will probably be will definitely be the most important lipid that we're going to be talking about for the rest of the year. It'll come back in the cell unit, it will come back in the cell transport unit, and it will come back in the photosynthesis and respiration unit. So just going back a minute, these kinds of fats are for long-term storage of energy. So when we eat too much sugar, our body converts it into fat and stores it in our body for the long term. Now, this isn't always a bad thing. I mean, it's more often than not bad to store too much fat now, but remember when we were hunter-gatherers and we weren't sure when we were getting our next meal, having a long-term supply, of stored energy in case there was a time where we weren't eating for several days, it would have been very helpful to have these kinds of fats. But the fats I'm about to show you have a different role in biology. So phospholipids have a phosphate group at the head, and instead of having a glycerol with 3 fatty acid tails, they have a glycerol with 2 fatty acid tails. So you're always going to look for the phosphate group, a glycerol, and 2 tails. Now remember that you're going to be given a diagram perhaps that you have never seen before on a test. So, just keep in mind that a phospholipid can be turned in space this way, it can be flipped that way. So you can't try and memorize one diagram. So you're going to be looking for a phosphate group, a glycerol, the 3 carbons in a row, and 2 tails instead of 3. So there is a hydrophilic, which means water-loving head, and by hydrophilic we mean chemically attracted to water. And the tails are hydrophobic, and they are repelled by water. And these part are nonpolar, whereas the head is polar. These hydrophobic tails are what make us not dissolve when we go into the shower, because our cell membranes are made up of hundreds of thousands of these phospholipid molecules all together. So, because you have a hydrophilic and a hydrophobic end, it makes lipid bilayers form. So you end up with lipids forming 2 layers, with the heads on the outside near water and the tails facing inwards towards each other, because you've got to consider that, if this is a cross section and if this were part of a cell membrane, there would be water on the outside of the cell. There would also be water on the inside of the cell. So this is referred to often as a phospholipid bilayer. Now, a cell membrane has other molecules in it. This is way oversimplified with just the phospholipids. There also are proteins in them. So here are 2 other pictures of phospholipids, just to sort of get you used to seeing different kinds of diagrams. In this one, they're showing you where the double bonds are. They are not showing you any of the carbons or hydrogens, because any biochemist just assumes they're there and knows they're there. So you can see the phosphate group, and here's where the glycerol is, and they're not showing you the carbons, but they're there, and then your fatty acid tails. And so we have our phosphate group, that's the polar or hydrophilic head, and those are the hydrophobic tails. So this actually is showing you a cross section. So if we had a sphere that would be very similar to a cell cut in half, you can see that the water would be attracted to the outside and the inside, and these tails, which are repelled by water, flip away from the water, so that there is a layer of hydrophilic heads in between the water and the tails. So there's 2 layers of tails next to each other, touching, and then the inside has got another layer of hydrophilic heads, and then there would be water inside the cell. And scientists actually believe that the first cells ended up developing in the ocean by just, if you ended up with phospholipids, they naturally form this lipid bilayer in these spheres called liposomes, and if they ended up trapping chemicals on the inside, that could have been the first primitive cells.